Hello students, welcome to this lesson. In this lesson, we are going to talk about herbicide resistance. Herbicide resistance refers to the ability of plant population to survive and reproduce despite being exposed to a herbicide that would normally kill the plant or severely inhibit its growth. This phenomenon is a significant challenge in agriculture and environmental management, as it can lead to reduced crop yields and increased cost for farmers. Some of the safest and most effective herbicides are such as glyphosate that kill any green plants. So here glyphosate cannot be used on crops because they kill the crop plants with the weeds. If we want to use glyphosate to kill weeds without affecting crop plants, we need to genetically modify crop plants to make them glyphosate resistant. So, a gene for enzyme that breaks down glyphosate can be introduced into a plant cell culture. For this purpose, we need to use tumor-inducing or TI plasmid of Agrobacterium tumefaciens to introduce glyphosate resistance gene into plants. One way to introduce transgenes into plants is to use Agrobacterium tumefaciens that has TI plasmid which causes tumors in the plants. The glyphosate resistance gene is inserted into the TI plasmid along with antibiotic resistance gene. The construct or recombinant DNA or recombinant plasmid is then reinserted into Agrobacterium tumefaciens. Plant cells are then exposed to transgenic bacterium and cultured on a plate containing antibiotics. In this case, only plant cells that have taken up the plasmid grow and the other are killed by antibiotics. As we can see from the diagram, glyphosate resistance and antibiotic resistance gene must be inserted or ligated in TI plasmid of Agrobacterium tumefaciens. This recombinant plasmid inserted back into the bacterium, then cut the leaf and chop up the leaf into smaller fragments of plant tissues. Expose the leaf tissue to bacteria carrying glyphosate resistant gene and antibiotic resistant gene. This allows bacteria to deliver the genes into leaf cells. Then expose leaf to an antibiotic to kill cells that lack the new genes. Wait for surviving cells to multiply and form a clump or callus. Here, one thing I need to remind you is antibiotics like kanamycin and hygromycin are commonly used in plant transformation as a selective agent to genetic modification of plants. This inhibits plant growth by affecting mitochondria and chloroplasts. This is the reason that glyphosate resistance gene is inserted along with antibiotic resistance gene. In this case, plant cells that acquire glyphosate resistance and antibiotic resistance gene survive in this antibiotic medium, allow callus to sprout or grow shoots and roots. The plants are then transferred to soil where they can develop into fully differentiated adult plants that are glyphosate resistant.